Everyone, let's get started. I think I know most everyone here in the audience already, but if I don't know you, my name is Trey Ideker. I'm a professor here at UC San Diego in the School of Medicine. I'm also, important for this conference, a co-director of the Cancer Cell Map Initiative project, along with Nevin Krogan here from UCSF. Wave, Nevin. He's got a very interesting network biology shirt on here. You can check it out later. This is the second time the CCMI, Cancer Cell Map Initiative, has organized one of these cell mapping symposia. The first annual one, was at the Gladstone Institutes up at UCSF last fall, so this time last year. And we hope that this dialogue around cell mapping will continue for many years to come, and really, as I think you'll see today, is just getting started. So what, first and foremost, is this notion of cell mapping? It's a two-word moniker for this conference and very general, but I think the premise of cell mapping is something that is going to be familiar to essentially everyone in the audience and is quite simple. It's the notion that if you look at complex diseases, by that very word complex, that means those diseases are caused not by a single locus or genetic variant, but combinations of loci, and probably not one combination, but many different combinations of variants, all of which give rise to a similar phenotype, that given disease, separating it from normal matched controls. By nature of that complexity, it's hard to think of even a single complex disease. Maybe someone can raise their hand if they can think of one for which we've mapped or think we understand more than just a minority or a fraction of the variants that are causal for that disease, much less the interaction between those variants and the environment and things like that. So what is then the sort of standard way these days in which people try to understand disease genes? I think, again, as most of us know, it's this procedure known as genome-wide association studies, or GWAS. And with the sort of rise and falling cost of genomic sequencing now, one can map for common diseases tens of thousands of patient genomes, hundreds of thousands of patient genomes, if that number of patients are in fact available with that said disease, along with matched controls, and then walk down the genome locus by locus in these Manhattan plots. So this is one end of the genome with chromosome 1 down to the other end of the genome, and look for correlations between the state of each individual genetic variant and the case control state of that patient over the patient population. And so in these Manhattan plots, sometimes you get lucky, or you do see a reasonably or a moderately sized effect. So you get one of these peaks representing a significant linkage between a disease or a locus and the disease. But much more typical of these kinds of studies is the plot over here on the right, where you get many suggestive associations at the weak strength of association, but none of which, or few of which, meet this genome-wide level of significance that's required because you're doing so many hypothesis tests as you march along. So that said, if one begins to look, and this is really the premise of cell mapping now, if one begins to look not at this level of individual variation or even how those variants map to genes, but if one now begins to map these variants at a higher level of cell biological information in pathways and networks and organelles, one can occasionally see structure that's quite common and quite strong, integrating all of those different rare weak variants. So for instance, if we have, in this obviously toy example, 3 different loci, one in blue, one in green, one in orange, each of these here has a very weak association, maybe suggestive association with the disease. And then I give you the added piece of information that, oh, these 3 loci are near genes that encode subunits of the same protein complex, or even genes within the same organelle, or some extra biological information about the structure of the disease in terms of cell and tissue biology, then I can begin to connect the dots and now test for association not for any one of these individual variants, but for the complex as a whole and variants in it versus the disease. And that can be quite powerful in terms of strength of association and in understanding the architecture of the disease. This is by no means a new idea. In fact, this idea that we should integrate and aggregate variants into pathways is an idea that's been used very successfully over genome projects for the past 5-10 years. I could have chosen essentially any of the cancer genome atlas mapping projects that have been published over the past 5 years, and there's at least one figure in those papers that show a pathway exactly with the intention of integrating variants. So for instance, here in this head and neck tumor genome paper from the TCGA, published a number of years ago now, they have organized here genes sort of largely into 5 pathway categories. Those are the columns here. So RTK, RAS, PI3K is the first pathway category they've drawn here. Cell death pathways are another category. But the point is important here. The general point is that variation hits each of these genes in tumor genomes typically quite rarely. 5% of HRAS, or 5% of head and neck cancer patients have a mutation in HRAS. But then if we realize HRAS is part of a larger pathway, then we're looking at 62% of patients which have mutations in that pathway. So this idea is by no means new. It's a classic idea in a sense, but there are 2 at least broad challenges that the CCMI Institute is trying to address. So one, I think experts would agree, those of us in the audience I think would agree, that pathway space, unlike the genome where we've covered, one can argue if it's 100% of the human genome that you actually report with the full genome sequence, but nonetheless, most of the human genome gets sequenced. We cover that space now with Illumina sequencing. But pathway space is very different. So pathway space, I think we'd all agree, it's ill-characterized how much of pathway space for a given disease has been covered, but it's probably the minority. So we first of all have to mount systematic efforts, just like have been mounted to sequence the number line of the human genome, now to map pathway space and really talk about what that means. And the second broad challenge here that we've outlined is even as information begins to accrue in these public maps, how do you then use those bits of information or in some total of information to translate the human genome, find disease genes, subtypes, and therapies? So that's the premise of the CCMI, and that was funded as an as a project. As one of these NCI Centers for Cancer Systems Biology about 1.5 years ago. Now, I'm very pleased to announce that similar efforts by overlapping investigators, not always the exact same investigators, have now been funded for 2 other main disease categories. So in addition to the Cancer Cell Mapping Initiative project, we just have seen funding of this Psychiatric Cell Mapping Initiative, and now, most recently, the Host Pathogen Interaction Mapping Initiative. And of course, all 3 of these initiatives share common core infrastructure and strategy, as I just talked about, and they're supported by this UC Axis and the QBI Institute up at UCSF. So without further ado, let me now move to the speakers. And I think you're going to find that the way this conference has been organized is exactly around these 3 disease foci, or people who really speak to this middle technology component of what is cell mapping. And the first speaker among this esteemed set of colleagues is Dr. Emma Lundberg. She's one of the directors of her version of cell mapping that's called the Human Cell Atlas Project, and she comes from Stockholm. So please welcome Emma. Thank you.